Good afternoon, everyone. It's Dina and Jaramany from the MiCA lab at KU Leuven in Belgium. I work on the AAA Code project, and today my talk will be under the title From Biomass to Barrier, Antimicrobial Properties of Functionalized Nanocellulose. Here's an overview of today's talk. I'll start by explaining the urgent need for antimicrobial coatings. Then I'll move on to the development of nature-inspired coatings. Then I'll present the objectives and methodology of our work and discuss key results, and finally wrap up with conclusions and future directions. We are constantly surrounded by high-touch surfaces. Think of handrails, doorknobs, hospital equipment, light switches, and even tables, like the one we have in this meeting room. According to the British Columbia Centre for Disease Control, more than 80% of common infections are spread by hands touching contaminated surfaces. And 16 million death cases per year are related with different infections, where the source of these infections is microbes inhabiting frequently touched surfaces. And although cleaning removes microbes from surfaces, however, common cleaning methods have no lasting effect, and they do not eliminate the risk of infection. That's why there's a pressing need for solutions that provide lasting protection, which is the need for antimicrobial coating. According to Market Analysis Report from 2023, as we can see here, silver, copper, and zinc-based compounds make more than 50% of commercial coatings. While they are effective to some extent, these approaches can face issues, like bacterial resistance, in which initially most bacteria are killed by the treatment. A few naturally survive and continue to multiply. Over time, these survivors acquire resistance. Eventually, they can form a protective biofilm, making them even harder to eliminate. And there are also some concerns about toxicity. So here comes our approach, which is the development of nature-inspired antimicrobial coating. Our approach is bio-based, it builds on biomass, and more specifically on nanocellulose. So the aim is to develop a nanocellulose barrier against infections. You might ask, why nanocellulose? Because it's bio-based, ecologically friendly product, and to date there are no significant bio-based antimicrobial coating on the market. It has functionalization possibilities due to the ample hydroxyl groups on the surface. It is safe, it is highly abundant, it has good mechanical properties, it's renewable and biocompatible. We just need to enhance the antimicrobial activity of the nanocellulose. Let me first introduce you to the three types of nanocellulose we work with. We have CNC, CNF, and BNC. CNC, which stands for cellulose nanocrystals, which are the least fibrillated among the three. They're rigid, needle-like particles with a diameter of about 5 nanometers and length between 20 and 100 nanometers. We receive CNC from our partner Melodia, which is the second largest CNC producer worldwide. Then we have CNF, which stands for cellulose nanofibrils, which are more fibrillated than CNCs. They form flexible fibers and can create 3D networks. Their diameter ranges from 5 to 60 nanometers, and we receive them from our partner SAPI. And finally, we have the BNC, bacterial nanocellulose, which is the most fibrillated among the three. It forms dense, spaghetti-like cross-linked network. Its diameter ranges from 20 to 100 nanometers, and we receive it from Cellugy. And CNC and CNF are produced from wood pulp. However, BNC is produced by biotechnological fermentation. This is how we see the nanocellulose in the lab. CNC has a transparent-like appearance. CNF and BNC are whitish in color, and they are more viscous than CNC. Because nanocellulose itself does not have an antimicrobial activity, we modify the nanocellulose either with tyrosidins developed by Stellenbosch University or with the 2AI compound developed at our lab, the MiCA lab, in collaboration with KU Leuven, and see how they inhibit potential pathogens. We start with the tyrosidins, which are a class of small cyclic antimicrobial peptides isolated from soil bacteria. They have a broad spectrum activity and multiple targets, and they have a rapid mode of action, which makes them less prone to resistance and highly stable. That's why they are great candidates for our coatings. Then we have the 2AI, the 2-amino-imidazole moiety, which is the active part of anti-fouling molecules produced by various marine sponges. That's why it's nature inspired. It has a dual activity. The first is an anti-biofilm activity in which it prevents the production of the slimy biofilm matrix, and it's a public good inhibitor. It selects against resistance. So if microbes acquire resistance to this inhibitor, they produce the slime again, and they will share this slime to the benefit of the susceptible microbes around. However, this production carries a metabolic cost, and these microbes do not, however, bear the energy cost of slime production. Therefore, they experience a relative benefit, and they outcompete the resistant microbes. And the other activity is the bactericidal activity when used at higher concentrations. For the modification of the nanocellulose with the 2AI, nanocellulose first undergoes tempo oxidation to form carboxylated nanocellulose, and then the negatively charged carboxyl group of the carboxylated nanocellulose interacts with the positively charged amino group of the 2AI to form the modified nanocellulose by ion-ion interaction. You might ask, why make it a coating? Why don't we just use the actives? It's for the durability and stability of the coating, also for the versatility of the coating, making it useful across very different environments, and also for the controlled activity and sustained activity of this coating, ensuring its long-term efficacy. And last but not least, the safety of this coating. So in order to develop this coating, we collaborated with 12 other partners, and here is us on the project, in order to develop a safe and bio-based antimicrobial coating. The objective of this collaborative work is to first give nanocellulose antimicrobial properties by binding our compounds to it, or by patterning, as Zhao explained in his presentation, and then to coat this nanocellulose to model substrates, plastic, glass, metal, and fabric, as Alexandra explained in her presentation, by incorporation of adhesion linkers. Then we will develop a spray coating or a thin film coating of this product, and then we will demonstrate it in a real bus environment. And finally, we will assess the life cycle of this antimicrobial coating. So first, to functionalize this nanocellulose, we modify it and test for its activity in suspension. And then we test the modified nanocellulose on coated substrates and see how it inhibits potential pathogens present on high traffic surfaces. So first we have the substrate, then we have the adhesion linker, and then we have the modified nanocellulose in order to form, in this case, the AMP functionalized nanocellulose coating, and also the same to have the 2AI functionalized nanocellulose coating. So for the evaluation of the antiplanktonic activity of this of the modified nanocellulose, we use this protocol. In a 96 well plate, we add our functionalized nanocellulose, then we add our bacterial culture 1-to-1 ratio, 100 microliters of nanocellulose, 100 microliters of bacteria. We incubate for 24 hours at 37 degrees, and then depending on the activity of our compound, if it has a strong antibacterial effect, we see killing of bacteria and reduction in the number of cells. And if it has a low antibacterial effect, we see growth of cells and increase in the cell counts. And this is quantified by plating and comparing to the growth control where bacteria grow normal. So we tested three types of nanocellulose, CNC, CNF, and BNC, modified with the AMPs and the two AIs at different concentrations, and based on our results, the most significant activity in suspension was CNC modified with the AMP at a specific concentration, and CNF modified with the 2AI at a specific concentration. So we used these combinations to proceed to work package 2, which is the assessment of the antimicrobial activity of the modified nanocellulose on coated substrates. We used this protocol for this assessment, in which on a 1 by 1 centimeter coated coupon, we add a droplet of bacterial culture diluted in TSB. We incubate for 2 hours at 37 degrees. We plate out the planktonic cells. Then we scrape, homogenize, and plate out strongly adhered cells. We tested our coatings against two pathogens, E. coli, because it is highly standardized, non-pathogenic lab reference. It offers comparative value, and Staphylococcus aureus, because it is also a standard reference for disinfectant and antimicrobial testing, and it is highly relevant for infection and control studies. First, we evaluated the activity of nanocellulose functionalized with the 2AI on coated glass against these pathogens, where we tested CNF on glass, CNF modified with the 2AI on glass, CNF modified with the 2AI with linker 1 on glass, and CNF modified with the 2AI with linker 2 on glass. And here we could see total inhibition of planktonic and strongly adhered cells on the coatings of modified nanocellulose compared to unmodified nanocellulose. We also saw the same activity against Staphylococcus aureus, which is the inhibition of all the planktonic cell counts and the strongly adhered cells compared to the unmodified control. Then we evaluated the activity of nanocellulose modified with the tyrosidins, also coated on glass, against these pathogens. Here we tested CNC coated on glass, CNC modified with the AMP with linker 1, CNC modified with the AMP with linker 2, also both coated on glass, and we compared also to a commercially available antimicrobial surface, which is called liquid guard. And here we could see a significant decrease in planktonic and strongly adhered cell counts on the CNC modified with the AMP with linker 2, and we didn't see this activity for CNC modified with the AMP with linker 1. And the results were also consistent against Staphylococcus aureus, where we only saw significant decrease in cell counts for CNC modified with the AMP with linker 2. So, to sum up, we had significant antibacterial activity of the AMP functionalized coatings against E. coli and Staphylococcus aureus for CNC modified with the AMP just with linker 2. However, we saw this significant activity of the 2AI functionalized coatings with both linkers, linker 1 and linker 2 for CNF modified with the 2AI. So for next steps, we will test the activity of multi-layer nanocellulose coatings with better mechanical properties. Then we will develop a spray coating and we will test it in a real bus environment. Finally, I would like to thank the organizers of the workshop. I would also like to thank everyone at the MiCA lab, especially my promoter, Professor Hans Stinakers, and my supervisor, Ines Stiers. And I would also like to thank everyone on the AAA Code project for the collaboration and communications. And finally, I would like to thank the European Union for the funding. Thank you.